Hello. So today we're going to focus briefly on hemoglobin, its structure, and then very shortly talk about the loading and unloading process of oxygen when it comes to hemoglobin. So the main LOs of today, as I mentioned before, is to describe the structure and function of hemoglobin, and then explain the terms loading and unloading of oxygen. The key terms for hemoglobin topic in general would be hemoglobin, quaternary structure, affinity, loading and associating, and then unloading and dissociating. Today, we're going to focus mainly on the top two and the last two definitions. So, to really start off this lesson, we should define what hemoglobin is. So, hemoglobin is a globular protein present in red blood cells, and its function is to carry oxygen from the lungs to the cells present in the periphery of the body via the blood. That is very wordy, but we can break it down. So, to discuss hemoglobin, we notice that it's a protein. And like all proteins, they need a very specific structure to function properly. So today, we're just going to kind of describe and talk about the structure of hemoglobin. So, start from the primary structure. Now, primary structure, as you might maybe seen before in previous lessons or topics, is the simplest form of a protein. Essentially, when DNA codes from amino acids, those amino acids can then combine and link together to form a polypeptide chain. That's essentially primary structure. Now, hemoglobin contains 4 polypeptide chains, which we'll make a note of here. So 4 polypeptide chains make up the structure of hemoglobin, as you can also see in the diagram on the screen. Secondary structure is when a polypeptide will then coil onto itself, in this case to form a helix. And I'll just draw maybe a simplified version of that here. Tertiary structure is when polypeptide chains change shape again onto themselves to have a more specific structure. Now, this is important in all proteins, as all proteins need an actual specific structure to function properly. So maybe draw another example here. I will just imagine that this is a very specific structure for hemoglobin. Now, the main diagram in the middle would be the example of a quaternary structure. Now, the main feature of quaternary structure is that it's made of more than one polypeptide, and it also may contain other groups. Now, when it comes to hemoglobin, the main points here is that these then 4 polypeptide chains then come together to form almost a spherical shape, as you can see here. This is important in the structure and the function of hemoglobin. Not only that, but in quaternary structure, they may also contain non-protein groups, which are called prosthetic groups. In this case, it would be these black dots you can see in the diagram. Now, these are heme groups. So each of our polypeptide chains are associated with one heme group. This heme group contains a ferrous ion. Ferrous ion just being an Fe2 plus ion. Now, the presence of this Fe2 plus ion means that we can now combine oxygen with hemoglobin. Just to recap, so each of these 4 polypeptide chains contains one heme group, the heme group containing iron. This iron can allow us to bind with oxygen. As a result, one hemoglobin molecule can combine with 4 oxygen molecules because it has 4 polypeptide chains, each of those chains containing a heme group. Now, this is important and it comes up quite frequently in exams. And if you were to perhaps scroll down, you'll see an example question of what you might be asked in an exam surrounding maybe the structure of hemoglobin. So we're given here a picture of hemoglobin. We see the 4 polypeptide chains, we see the heme groups and oxygen binding site. We also are then asked, what's the evidence here that from the diagram that hemoglobin has a quaternary structure? Now, if we go back to remember what we said in the beginning, quaternary structure means there's more than one polypeptide chain involved in the structure of this protein. Now, quaternary structures may have these non-protein groups, in this case a heme group, but not always. And so this exam question is really testing your core knowledge of your protein structure. As a result, the answer here should be there's more than one polypeptide chain present. Yep, I will see here that is the answer. And it says here as well, ignore references to heme or other groups. And it said this because the main point of quaternary structures is that there's more than one polypeptide chain. So quaternary structure may have another group associated with it, but not always. And so that's basically testing our core knowledge of our protein structures. So, just to recap, hemoglobin is made up of four polypeptide chains, each of which is associated with heme groups which contain ferrous iron, and that iron then allows us to combine with oxygen. As a result, hemoglobin can carry up to 4 oxygen molecules. And so that overall does really encompass the structure of hemoglobin. Now, we're going to very briefly look upon the loading and unloading of oxygen when it comes to hemoglobin as well. So, for hemoglobin to really carry oxygen, it needs to take on oxygen and also offload the oxygen to cells that need it. And so where this takes place is also important. So I'm going to briefly write upon here where loading and loading takes place of oxygen in hemoglobin. So loading takes place in the lungs, and unloading takes place in maybe in our peripheral cells, let's say our muscle cells. So if we go back to maybe biology and ventilation, we know that ventilation takes place in our lungs. And so in our lungs, there's a very high concentration of oxygen. And that's where our red blood cells take on oxygen. And this is where hemoglobin becomes important. And so loading is basically the process of hemoglobin binding to oxygen. And this takes place, as I said before, in the lungs. So if I maybe draw out these blue dots, oxygen, this red blood cell can now take up this oxygen due to the presence of hemoglobin. This red blood cell might then travel across the body through our circulatory system, so throughout our blood vessels, carrying this oxygen molecules to whatever cells that need it. Now, our muscle cells in this example would be a good way of describing how oxygen is needed in our certain cells in our body. For example, if we're undergoing rigorous exercise, our cells need to undergo respiration. And for respiration, we need oxygen. And so what happens here is that hemoglobin can then dissociate with these oxygen molecules, allowing it to our cells to use it instead. And that's what we call the unloading / dissociating of oxygen, essentially, when hemoglobin unloads its oxygen to our body cells. And so this is a very brief diagram of how loading and unloading oxygen in our red blood cells works with the help of hemoglobin.